Good day, dear students. Today's my lecture will be about another system of organs, male reproductive system. And during this lecture I will tell you about the main organs of the male reproductive system. But first of all about its development. The development of gonads arise as thickening of mesodermal epithelium, the genital ridges, on the mesial surface of the mesonephros. Please look on this scheme. Here you can see the indifferent stage of the development of the reproductive system. So mesial wall of the mesonephros forms the thickening which get the name genital ridges. These ridges are formed by the small cuboidal epithelial elements and scattered between them the sex primitive cells. Look here, this is the genital ridges, and here you can see the primitive sex cells, gonoblasts. Main epithelial cells in these genital ridges, develops from the mesenchyme here in situ, and these epithelial cells penetrate the lining mesenchyme to form the sex cords. Long structures are sex cords. But primordial sex cells are migrants. They come here from the yolk sac endoderm, and here they develops. Then they pass by the blood vessels from the yolk sac to the pelvic region of the trunk of the embryos and invade these sex cords. And this scheme show you that during this indifferent stage present two parallel long ducts, mesonephric duct and parallel to him paramesonephric duct. Genetic sex of the embryos is determined at fertilization by the absence or presence of Y chromosome. Gonadal sex is determined by the presence of SRY gene, special gene located in the sex-determining region, which locates on the short arm of the Y chromosome. A specific DNA-binding protein called the Testis determining factor, international abbreviation TDF, encoded by the SRY gene, has been found to be directly responsible for the testicular development and differentiation. So, if embryos has Y chromosome, you know that it is male organism. If Y chromosome absent and present two X chromosomes, it is the genetical sex of the female. That's why during the first 7 weeks of gestation, it is impossible to say what will be the sex of the future baby. Both sexes has in different stage of the development of the reproductive organs. But after 7 weeks of the gestation, the sex of the embryos become recognizable. And in this case, if it will the way of the male reproductive system, the sex cords become more distinct and elongated and form the seminiferous tubules. Their peripheral ends anastomose and unite with a number of mesonephric tubules to form the rete testis. The second source of the development of the testis is mesenchyme. Mesenchymal cells separate the seminiferous cords from one another and give rise to interstitial cells, so-called Leydig cells. And these cells develops very early between the seminiferous tubules and start to produce testosterone. And testosterone stimulate development of the indifferent primordium into a testis. The second very important event takes place when the Sertoli cells developed. Sertoli or sustentacular cell. These cells develop within the seminiferous cords, and these cells produce another very important hormonal substance called Mullerian inhibiting factor, international abbreviation MIF, Mullerian inhibiting factor. The further development of the testis and external genitalia, also from the sexual indifference stage, occur at the same time and result from the action of dihydrotestosterone. It is modification of simple testosterone, international abbreviation DHT. That's why the function of DHT, simple testosterone, and MIF lead to the development of the accessory glands, genital ducts, and other and external genitalia of the male organism. Very interesting that paramesonephric duct obliterates, but mesonephric duct, which get the name Wolfgang duct, undergo the further development under the influence of this dihydrotestosterone and Mullerian inhibiting factor. So, if Y chromosome absent, no SRY gene, no sex-determining factor, in this case, the development of the indifferent stage of the sex organs will go into the way of female reproductive system. And in this case, mesonephric duct will obliterate, but paramesonephric Mullerian duct will get the further development. But in male reproductive organs, the mesonephric duct is transformed into the ductus epididymis and ductus deferens. Connection of the ductus epididymis with the rete testis is established by a number of mesonephric tubules that become the ductuli efferentes. The paramesonephric duct almost all duct obliterate, involute, and sometimes leave only small rudiments. So the male genitalia system you can see on this scheme. So the main gland of male reproductive system is testis. It is paired organs which locates outside of the pelvic region of the trunk and is covered in the scrotum. It locates in the scrotum and covered by 3 tunics. Outermost tunic, middle, and innermost or internal tunic. Then capsule send septors which divides the testis into compartments or lobules. You see, compartments and or lobules. And also here as part of capsule appear the connective tissue which we call as mediastinum. And here the highly coiled seminiferous tubules from the compartments meet and change into the straight tubules and meet the rete testis. I told you one moment ago that it is the branches of the mesonephric duct. So form the rete testis. Then tubules of the rete testis go into the ductuli efferentis. You see ductuli efferentis, they are numerous, and they go to next organ of the male reproductive system. It gives the name Epididymis. Epididymis locates on the posterior surface of the testis. It is flat and long organ. So it has head. Head mostly consists of the ductuli efferentis. Each tubule opens into the ductus deferens, which travel from the head of the epididymis to the body and tail. And body and tail of the epididymis mostly consist of highly convoluted ductus epididymis. Then ductus epididymis leave the epididymis and change into the ductus deferens. Ductus deferens forms the small descending part and then long ascending part. And ductus deferens go to the pelvic region of the trunk, forms the dilation ampulla region, and then opens into the urethra. Urethra and ejaculatory duct in male reproductive system, it is the same. Besides these organs, male reproductive system has 3 pairs of accessory glands. The first gland, it is seminiferous vesicles, seminal vesicles, then prostate gland and bulbo-urethral glands. We call these structures as glands, but in reality, glands are also bulbo-urethral glands. Seminal vesicles, it is highly coiled ductus deferens, which locates in the tunica mucosa and tunica muscularis of the ductus deferens. And prostate gland, it is complex of many 30 to 50 up to 50 small tubular alveolar glands, which directly opens into the posterior regions of the ductus deferens and beginning of the urethra. You can see that this gland, prostate gland, it is not one large gland. So now I'll tell you about testis. Now, as you saw, the testis is a double gland, and functionally it is dual gland. it is both exocrine and endocrine. This gland belong to the peripheral and endocrine system because of presence of the clusters of the endocrine cells in the stroma of the organ, which produce the main male sex hormone, testosterone. It is the function of the endocrine portion. Exocrine portion, its principal function, the production of spermatozoa. Testis is suspended within the scrotum and is immediately surrounded by the testicular capsule. It has three layers, I told you. Outer layer, tunica vaginalis, middle layer, tunica albuginea, and innermost layer, tunica vasculosa. You can see the outer tunic has two layers. It is tunica vaginalis parietal layer, tunica vaginalis visceral layer, tunica albuginea, and innermost tunica vascularis. You can see the testis, mediastinum of the testis, and epididymis on the posterior wall of the testis, locates on its capsule. Thickening of the tunica albuginea, it is mediastinum of the testis. Also from this thickening started the septus, connective tissue septus, you see, which divides the organ into the regular triangle-shaped or pyramidal-shaped compartments or lobules. And inside each lobule or compartment present 1 to 4 highly coiled seminiferous tubules. They are blind ending on the periphery and closed into the top of the pyramid. They empty into the straight tubules, and then straight tubules forms the rete testis. It is a light microscopic picture. It is hematoxylinazine staining picture of the testis and epididymis. You can see the tunica albuginea. You can see the mediastinum with the red testis with the straight tubules. You can see the plates of the loose connective tissue septus, which divides the organ into the compartments. That's why in each compartment we can see the highly coiled seminiferous tubules, between them loose connective tissue, which we call it stroma. Stroma usually contain the blood and lymph vessels, nerve fibers, and also here we can find the clusters or islets of endocrine cells, interstitial cells or cells of Leydig. Usually these cells occurring in groups and they produce testosterone, male sex hormone. This light microscopic picture of high magnification, you can see the capsule and you can see the tubules. The diameter of each tubule is over 0.2 millimeters in diameter and 30 to 70 centimeters long in human. These tubules can anastomose with one another, forms loops, and sometimes anastomose even with the seminiferous tubules of the adjacent lobules, not only inside the concrete lobule, but also with the seminiferous tubules in the adjacent lobules. In the apical region of the pyramid, these straight tubule, these highly convoluted tubules change into the straight tubules. You see loose connective tissue between the seminiferous tubules and clusters of Sertoli, Leydig cells and acrine cells. So, wall of each seminiferous tubules is lined by the germinal or seminiferous epithelium. It is modified, striated, cuboidal epithelium. Like other types of epithelium, this epithelium also has a thin basal lamina. And under the basal lamina present the peritubular connective tissue, loose connective tissue with collagen fibers, cells of the loose connective tissue like fibroblasts, mast cells. And then the outer layer of these seminiferous tubules are myoid cells. It is modification of the smooth muscle cells, but these cells are not spindle shaped. They has processes. They are slightly stellate, and neighbor cells exhibit junctional complexes between neighboring cells. So this light microscopic picture after hematoxylinazine staining, here is high magnification. We can see the loose connective tissue. You see loose connective tissue between the sections of the seminiferous tubules. Now it is the sections of the seminiferous tubules. Between them is the loose connective tissue, stroma of the testis. In the wall of the seminiferous tubules, we can see these long cells with processes. It is myoid cells. And in stroma between the seminiferous tubules present interstitial cells. You see interstitial cells may be binucleated, very rare, but mostly has only one nucleus. Cells ovoidal and has a lot of lipid inclusions. They produce steroid hormone testosterone, which is lipid soluble hormone which synthesized on the base of the lipids. So Sertoli cells also present in the wall of the seminiferous tubules. So Sertoli cells are cells on the basal lamina of the seminiferous epithelium. Seminiferous epithelium, it is place of the production of the new sperm cells, and spermatogenesis takes place in the seminiferous epithelium of the seminiferous tubules, or spermatogenic epithelium, or seminiferous epithelium. These words are synonyms. So now you can see in details on this scheme the wall of the seminiferous tubules. You see the striated, thick seminiferous epithelium, basal lamina, then peritubular connective tissue with the capillaries, with many interstitial cells, and layers of myoid cells. Now you see one layer of the myoid cells. On the basal lamina of the seminiferous epithelium, special cells are located. These cells we call as sustentacular cells or Sertoli cells. These cells derive from the embryonic genital ridges, mesoderm. And these cells, it is light microscopic picture after hematoxylinizing staining. You see the lines cover the boundary of the, show us the boundary of the Sertoli cells. You can see that it is very tall cell which penetrate all the epithelium. Its apical part get the lumen. These cells, that's why are long, branched. They have processes, lateral processes. And this scheme is not visible. Here in this region mostly present processes. These cells has cytoplasmic enfoldings, and you see that spermatogenic cells occupy these enfoldings on the outer surface of the Sertoli cells. And spermatogenesis is impossible without Sertoli cells. Sertoli cells has very well developed smooth endoplasmic reticulum and Golgi complex, has numerous mitochondria and some rough endoplasmic reticulum. On the electron microslide, it is transmission electron microslide, we can see Sertoli cells in details. You see basal lamina, seminiferous epithelium, Sertoli cell which lies on the basal lamina and get the lumen. And the enfoldings on the surface of the Sertoli cells locates the spermatogenic cells. Here it is the spermatogenic cells. And these Sertoli cells, together with the peritubular connective tissue, forms the so-called blood-testis barrier. This barrier protects the developing spermatogenic cells from the blood antigens which present in the circulatory blood, in the blood vessels of the stroma around the seminiferous tubules. That's why blood-testis barrier protects developing sperm cells from the outer immune damage, from the outer immune attack. Pay attention, look on these lateral processes of the Sertoli cells. These processes, you can divide the seminiferous epithelium into two compartments. This scheme shows you in details the Sertoli cells and spermatogenic cells which locates on the cytoplasm on the surface of the Sertoli cells. So lateral processes of the Sertoli cells meet the same lateral processes of the neighbor Sertoli cell forms the tight junction complex with one another and divides the seminiferous epitheliums into two very regular compartments. Basal compartments between the basal lamina and the processes. And cells in this region, the components, you can see orange, are lined by the orange color. Then, adluminal compartment. It prolongates from the lateral processes of the Sertoli cells to the lumen. And spermatogenic cells which occupy these compartments are lined by the blue color. So this organization of the spermatogenic epithelium is very regular and is necessary for the successful spermatogenesis. If Sertoli cells damaged and these processes damaged, takes place the mixturing of the spermatogenic cells in the spermatogenic epithelium. In this case, the spermatogenesis stops. So, let's summarize the very important functions of the Sertoli cells in the seminiferous epithelium. Now, first of all, it gives the physical support for the spermatogenic cells. Very interesting, but spermatogenic cells pass 4 stages of its development, and cells in each stage are not independent. They attach to one another by the cytoplasmic bridges, and together forms the syncytium. It appears the huge mass of cells which are connected with one another by the thin bridge of cytoplasm. This physical support for this syncytium also is provided by the Sertoli cells. Then, second function, nutritional regulation of the developing spermatozoa. Also protection from autoimmune attack by the barrier I told you 2 minutes ago. Also phagocytosis of the residual bodies shared by the maturing spermatozoa. Later I'll explain you this position. Also Sertoli cells secrete fluid for sperm transport, including the androgen binding protein, ABP, which combines with the testosterone produced by interstitial cells, and also Sertoli cells produce hormone inhibin, which acts on the pituitary to decrease follicular-stimulating hormone production. And also Sertoli cells secrete some estrogens, female sex hormones. Why it is so necessary, the secretory function of Sertoli cells? New formed sperm cells are unmovable. They can't move themselves, and in testis present and in genital ducts present special mechanism for the pushing the new sperm cells in the distal direction to the ejaculatory duct. So it is one of the mechanisms of the pushing of the sperm cells. Next very important function of the Sertoli cells present in the fetal period, production of anti-Mullerian hormone, MIF, anti-Mullerian inhibiting factor, which helps in the development of the male reproductive system and obliteration of the paramesonephric duct, which is necessary for the development of the female reproductive system. You have to understand that if this hormone will not produced in necessary volume, in necessary concentration, will be abnormality of the prenatal development, which is called as hermaphroditism. Hermaphroditism means that newborn baby and then man will have the both organs, will have the sex organs of both sexes together, male and female. And it is a very hard situation for the person. And the last very important function that Sertoli cells synthesize and secrete testicular transferrin, an upper protein that accepts iron from the serum transferrin and converts it to maturing gametes. Maturing gametes also need ferrum or iron for its maturation. So now let's talk about the process which get named spermatogenesis. The germ cells comprise a stratified layer of epithelium 4 to 8 cells deep, lining the seminiferous tubules. The spermatogenic cells differentiate progressively from the basal region of the tubule to the lumen. The sequence of events is referred to as spermatogenesis, which involves the two processes of cells multiplication, including reduction from the diploid to the haploid number of chromosomes, and cellular differentiation, spermyogenesis. So I think that you know the stages of spermatogenesis. I repeated stages: first multiplication, then growth, then maturation, then formation or spermyogenesis. So spermatogenesis occurs about 72 to 74 days. It is cyclic process. If it starts, it will last 72 to 74 days, and then appear the mature sperm cells. But in each tubule, this process may be in different times. Not all seminiferous tubules together start spermatogenesis. In some tubules may be the first stage of multiplication, in another tubules may be the stage of the growth, in another stage of maturation. That's why the concentration of sperm cells in geniculate is approximately same during every day. But this process is cyclic and occupy different tubules. In different times this process start and continue also. Start this process from the deepest cells in the seminiferous tubules, which rest on the basal lamina. It is spermatogonia. Spermatogonia, it is the next step of the development after the gonoblasts, primordial sex cells. So spermatogonia locates also on the surface of the Sertoli cells in special imaginations, but only in the region of the basal compartment. And these cells usually are small, spheric, with the large nucleus, and they can divide methodically. And that's why they start the phase of multiplication. The first step of multiplication is beginning by the spermatogonia. Spermatogonia, it is the normal cell with diploid number of chromosomes, 44 autosomes and 2 sex chromosomes. If it is male, of course, sex chromosomes X and Y. So these cells divide methodically and slightly moves up and go to the next stage, to the growth phase. Second stage, growth phase. Phase of growth of spermatogonia. Spermatogonia really grow, enlarge, and become larger. You can see transformates into the primary spermatocytes. These cells also locates in the basal compartment of the seminiferous epithelium. These primary spermatocytes also diploid cells, very large cells, and live very short period of time. When they break the contacts between the processes of the lateral processes of the Sertoli cells, they pass from the basal compartment to the abdominal compartment. And then these tight junctions restore and again appear the regular architecture of the spermatogenic epithelium. Next, third phase of the spermatogenesis get the name phase of maturation. During the phase of maturation takes place the reduction division, which is called as meiosis. Primary spermatocytes divides by the meiosis, pass 2 phases of meiosis. During the first phase appear the secondary spermatocytes. Secondary spermatocytes are half of the primary spermatocytes, and now it is also diploid cells with 23 pairs of chromosomes. But during the first division of maturation takes place very important genetic process, crossing over. During crossing over, the homolog chromosomes exchange with the genes. That's why provides the variability of the genome of the mankind. So next stage, it is second meiotic division. And after the second meiotic division, secondary spermatocytes divides into 2 spermatids. Spermatids are oval-shaped cell with the oval-shaped nucleus, elongated cells, usually elongated cells, and they occupy the top of the seminiferous epithelium, the upper regions of the abdominal compartment. So it is also short-living cells, and these cells penetrate into the Sertoli cells. This scheme shows you the syncytium of the spermatogenic cells. you can see that start from the Spermatogonia of type A and B, then a mitosis of them, then appear the primary, secondary spermatocytes, spermatids. All cells has connection with one another. You see appear the mass of cells, which is called as true syncytium. And as a result of this process appear the mature spermatozoa. You see that spermatids, you see elongated oval-shaped cell change into the mature sperm cells. Mature sperm cells, it is very specific cells. As you remember, it has head, short neck, and long flagella. So, the last phase of the spermatogenesis, it is phase of formation or spermyogenesis, when elongated oval-shaped cell change into the mature sperm cell. And this transformation takes place inside the cytoplasm of the Sertoli cell. Spermatids penetrates into the Sertoli cells, and here in cytoplasm of Sertoli cells, it passes 4 stages of the spermiogenesis. These spermiogenesis include Golgi phase, cap phase, acrosomal phase, and maturation phase. You see that spermatid undergo very deep and serious changes. You see, first of all, it is oval-shaped cell with very well-developed Golgi apparatus, 2 centrioles, a lot of mitochondria. Then in Golgi apparatus appear the hydrolytic enzymes, and this Golgi apparatus transformates into the acrosomal vesicle. Golgi apparatus is filled by the hydrolytic enzymes like trypsin, acrosin, and hyaluronidase, and locates like cap on the nucleus, on the front surface of the nucleus. You can see that forms the flagella, and mitochondria. Next step, acrosomal cap appear acrosome, and mitochondria migrates to the flagella and locates around it, and appear the spiral allocation of mitochondria around the flagella. So, but you see that excess of the cytoplasm of spermatids is not necessary for sperm cell. That's why excess of cytoplasm is expelled from the cell like residual bodies, and cytoplasm of the Sertoli cell utilize these residual bodies. Lysosomes of the Sertoli cells utilize this excess of the cytoplasm of the sperm cells. So after these 4 stages appear the mature sperm cell. You see it configuration on this scheme. This scan electron microgram show you the mature sperm cell on the surface of the uterine cavity of the rodent. You can see the ciliated cells of the uterus, and on the surface of the endometrium present these very specific cells, head, short neck, and long flagella. But I pay your attention that in new formed sperm cells, flagella is unmovable, is not active. Some words about interstitial cells. Interstitial cells are shown in this picture. It is microslide, hematoxylinase stained microslide. You can see the part of some sections of the adjacent seminiferous tubules. You can see the stroma, loose connective tissue between the seminiferous tubules, and large ovoid or spheric interstitial cells or Leydig cells. You see that their cytoplasm is saxophilic, pale staining. Usually these cells locate in groups near the blood vessels, of course. Fenestrated blood capillaries is necessary for the passage of the hormone testosterone, which is secreted by these cells. These cells are very rich in lipid inclusions and smooth endoplasmic reticulum. Functional consideration of the testis. You have to understand that testis, it is not independent organ. This organ is regulated by the central endocrine system, and the production of testosterone and production of the new sperm cells are both regulated by the central endocrine system. During the first over 14-15 years, it is personal, of the lifespan of the boy and then man, the production of sperm cells and testosterone don't present. It starts only after puberty, when it activates the gonadotroph cells of the adenohypophysis. But these gonadotroph cells also are not independent and stimulated by the releasing factors of the hypothalamus, of the medial and posterior nuclei of the hypothalamus. So under the influence of the gonadotropin releasing factor start the production of gonadotroph hormones of the gonadotropic cells of the adenohypophysis, pars distalis. So these cells produce so-called luteinizing hormone, LH hormone, luteinizing hormone. This luteinizing hormone in male organism we call as interstitial cell stimulating hormone, ICSH, interstitial cell stimulating hormone. This interstitial cell stimulating hormone stimulates the activity of the Leydig cells in the interstitium. These cells which I showed you one moment ago. These cells produce testosterone, which control the spermatogenesis in the seminiferous tubules. And this testosterone activates the phases of spermatogenesis and appear the new clones of sperm cells. That's why you have to understand that the testis, it is not independent organ. This scheme show you my story. You see the hypothalamus, which produce gonadotroph stimulating factors, releasing factor, which has the influence target cell as gonadotroph cells on the anterior hypothalamus. This gonadotroph cell produce luteinizing hormone, which stimulate the Leydig cells in the interstitium of the testis. These cells produce hormone testosterone, which activates the spermatogenesis. Sertoli cells in the seminiferous tubules also has influence on the gonadotroph cell? I told you that it produces the hormone inhibin. Not in very low concentration, but it is so. So hormone inhibin decreases the production of the second hormone of the gonadotroph cells, follicular stimulating hormone. That's why Sertoli cell decreases the production of the follicular stimulating hormone, which is not fruitful for the male reproductive system. This hormone is very important for the female reproductive system. And next lecture we will talk about this. But in male reproductive system, follicular stimulating hormone is deactivates by the hormone inhibin produced by the Sertoli cells. Now some words about the male genital ducts. This is the system of tubules which helps the sperm cell to reach the ejaculatory duct. And so these genital ducts develops from the mesonephric duct, branches of the mesonephric duct. So first it is straight tubules which attach to the seminiferous tubules, and these straight tubules form the rete testis. Now you can see that together all male genital ducts function in the maturation, storage, and transport of spermatozoa. So straight tubules, which forms the rete testis, you can see on this section. It is a microslide which show you the transversal section of this rete testis. You have to understand that these tubules, all tubules, has the same plan of the construction of their wall. It is really hollow tubes, and their wall has the same principles of the organization. First of all, they are covered by the epithelium. Epithelium differ in different regions, but present everywhere. Epithelial cells rest on the basal lamina. Under the basal lamina present the loose connective tissue, peritubular connective tissue, and then smooth muscles. And contraction of the smooth muscles helps the sperm cells to pass down to the ejaculatory duct. So two mechanisms of the pushing of the sperm cells. First, I explained you the secretion of the fluid by the epithelial cells of the lining epithelium, and the second one, the constant contraction like waves of the smooth muscle cells in the wall of these ducts. So concretely in the rete testis, the epithelium is low cuboidal. They has two types of cells. Some of them are ciliated, and some of them with short microvilli. And this epithelium covers the straight tubules. You can see the peritubular connective tissue and smooth muscles in the wall. Next, ductuli efferentis. Imaging from the rete testis to form lobules of the epididymis. Ductuli efferentis are connected with the straight tubules of rete testis, and then leave the testis and go to the epididymis. And here in epididymis they form the head of the epididymis, and also in the head we can distinguish the lobules of the epididymis. Each ductule is about 6 to 8 centimeters long and about 0.05 millimeters in diameter. Under the epithelium covers this ductul efferentis, and under the epithelium is epithelium are bounded by connective tissue, and each is surrounded by a thin layer of circularly arranged smooth muscle fibers. On this microslide, stained by hematoxylinazine, you can see the section of the ductul, one of the ductul efferentis. So now in details you can see the epithelium covering epithelium. The epithelium here is simple column, but you can see that in this epithelium is very easy to recognize two principal types of cells. One of them are tall columnar cells. You can see tall columnar cells with cilia. You can see that these cells forms the group on the basal lamina, and they have long cilia. The second type of cells are low cells, cuboidal cells. You see low cells, cuboidal cells, and these cells has microvilli on the free surface, also form the group. That's why in this epithelium we can see the group of tall columnar cells with the cilia and the group of low cuboidal cells with the microvilli. It is very prominent sign of the ductul efferentis, epithelium of the ductul efferentis. Ductul efferentis, each ductule opens, unite with one another and forms the single ductus epididymis. This duct, which is surrounded by the connective tissue, is highly tortuous and forms the remainder of the head, the body, and the tail of the epididymis. So this epididymis travel from the neck up to the tail. They coiled many times, and here sperm cells also pass slowly by this ductus. So as other tubules, this ductus epididymis is covered by the epithelium, then peritubular connective tissue, loose connective tissue, and smooth muscle cells around. Pay attention to the epithelium. Here in epithelium is pseudostratified columna. Look here, pseudostratified columna. But here, if we compare with the ductul efferentis, the low cells get named basal cells. Low small cells are basal cells. They don't reach the lumen of the ductus. But tall columnar cells reach the lumen and has cilia. That's why now all cells looks like one another. They are tall columnar and has cilia, but between them present short basal cells. Ductus epididymis leave the epididymis on its lower part and empty into the ductus deferens. Ductus epididymis straightened out at its termination and becomes continuous with the ductus deferens. And the ductus deferens ascends from the scrotum to the inguinal region, traverses the inguinal canal. So this ductus deferens is very long. It has long ascendance tubule which get the trunk, pelvic region of the trunk, and here forms the dilation, ampulla region. So this light microscopic picture show you the section of the ductus deferens, and you can see that it is also covered by the epithelium, covering epithelium. Also, now it is also pseudostratified. Then a peritubular connective tissue. Together they form the tunica mucosa. Peritubular connective tissue now get the name lamina propria of the tunica mucosa. Then layer of submucosa, because this duct is comparatively large and has comparatively a thick wall. So under the tunica mucosa we can find the tunica submucosa, loose connective tissue around, and then muscular coat, not thin muscular coat. It consists of layers of the smooth muscle cells. And you see how thick is smooth muscles and tunic. And around is the tunica adventitia. It is also connective tissue. Pay attention on the epithelium. This pseudostratified epithelium has tall columnar cells with very long stereocilia on the apical surface. Beating of these stereocilia, it is the third mechanism for the pushing of sperm cells, unmovable sperm cells. These cilia, stereocilia, beat into the direction of the jejunculatory duct and also push the new formed sperm cells to the jejunculatory duct. So in the ductus deferens and jejunculatory duct, which now it is combined with the urethra, opens three accessory genital glands. These, first of all, seminal vesicles. Seminal vesicles are not really gland. It is tortuous elongated diverticulum of the ductus deferens at the termination of the ampulla portion and is situated posterior to the prostate gland. This is paired glands. Paired seminal vesicles are highly coiled tubular structures about 15 centimeters long. The lower portion of the vesicle becomes a narrow, straight duct that joins with the ductus deferens to form the jejunculatory duct. This picture shows you that seminal vesicles, it is really ductus deferens. It will consist of the external connective tissue, adventitia, with a lot of elastic fibers, smooth muscle coat, and mucous layer. And you can see that mucous layer forms very numerous long branched folds. You see, these folds may be primary, you see, primary folds, then secondary and tertiary folds. So these folds branched very much and appear this interesting picture under the microscope. And because of these numerous folds, numerous compartments of different size are formed. And epithelial cells, tall columnar epithelial cells, which covers each of these folds, produce secret. And this is the secret of the seminal vesicle. Look here at the larger magnification of the pseudostratified epithelium of the seminal vesicle. You can see epithelium, it is fold, and epithelium, which primary fold, secondary fold, and epithelium. We can see that columnar cells covers it. It has flagellum projection from the apical surface, and between them present the small short cells with the microvilli, which don't get the lumen. And very interesting that the secret of this gland is yellowish. Secret is viscid liquid. Next gland, prostate gland. It is the largest of the accessory glands, is pierced by the urethra and the ejaculatory duct. All this gland has capsule, comparatively thick capsule, which is richly vascularized, has dense regular connective tissue, thick bundles of the collagen fibers, and in the stroma we can see the smooth muscle cells. It is unicum. Only one gland in our body, but only in man bodies, has smooth muscle cells in the stroma. And also here present cells of the loose connective tissue. So if you will look on this scheme, you will see that the prostate gland, it is a conglomeration of 30 to 50 individual compound tubular alveolar glands, you see, which is arranged in 3 discrete concentric layers around the urethra. So in the center is urethra. Around, in the tunica mucosa of the urethra, present the mucosal glands. Then in tunica submucosa of the urethra, present the submucosal glands, and main group outside of the tunica submucosa. Each small gland has its own excretory duct, which opens into the urethra. It is light microscopic picture. This is heme, this is light microscopic picture after the hematoxylin staining. On the section is visible the urethra, which is covered by the transitional epithelium, and then groups of glands. You see in tunica mucosa, small groups of cells of glands, in tunica submucosa, and main group of glands. You see, between this group of glands present the smooth muscle cells. Why these cells are necessary, smooth muscle cells? No, it is larger magnification of the group of glands of the prostate gland. And you can see very clearly the smooth muscle cells around the secretory portions of these glands. Why it is so necessary? These glands don't expel secret all the time. It produces secret and collect it into the secretory portions and into the ducts of this gland. And during ejaculation, these smooth muscle cells together contract and push the secret of this gland into the ejaculatory duct. And especially the main volume of the ejaculate is produced by the prostate gland. The volume of ejaculate is over 2-3 milliliters. It is mostly the secret of the prostate gland. So, very interesting that epithelium, which covers the secretory portions, lined by, it is simple to observe the stratified columnar epithelium. The main component of the prostate secret, it is proteins, and that's why the epithelial cells has very well developed rough endoplasmic reticulum. Golgi apparatus has numerous secretory granules and lysosomes. And very interesting that aging of the person is accompanied by the formation of the prostatic concretions or corpora amylacea. You see corpora amylacea, here it is oxyphilic structures which looks like small bones. They locate inside the secretory portion. You see, inside the secretory portion. The number of these concretions increase with the age, person's age. But the significance of these concretions is not understood today. Now you can see the section, large magnification section of the human prostata. You can see the secretory portion, cells, columnar cells, some of them short cells. You can see the smooth, very well see the smooth muscle cells between the secretory portions of the placenta. A lot of blood vessels also in the loose connective tissue around the epithelium. You see the basal lamina of the epithelium. And the last glands are bulboretral glands. Now it is really glands. The second name, glands of Cowper. It is paired bodies, each of size of pear, lying in the connective tissue behind the numerous membranous urethra. Really, it is small glands. It is a compound tubuloalveolar gland whose long duct drains into the proximal part of the penile urethra. The bulboretral glands is surrounded by a thin connective tissue capsule, external to which are skeletal muscle fibers. And so the epithelium of these glands also simple, consists of cuboidal and columnar cells. Secretory ducts are covered by, are lined by the pseudostratified epithelium too. So secretion is clear, viscid and mucus, and is rich in cellular proteins and amino sugars. The secret of bulboretral glands, it is also the component of the ejaculate. So, doctors, thanks for your attention. I described all organs of the male reproductive system. Our time is over and you may be free. Goodbye.